Hello students, and welcome to Unit 11. This is a unit on examining the types of growth habits and cell division habits that various types of microbes have. And in this lecture we will also discuss various laboratory techniques that you would use if you were working in a microbiology laboratory. And most of these techniques that I'm going to discuss apply to bacteria. Most of them apply to bacteria. I discussed the methods for handling viruses in the virology lecture, in the previous lecture. So for the most part, what I'm going to talk about here is growth and analysis methods for bacteria. And what I'm going to say here applies a little bit of it applies to fungi, and a very little bit of it applies to protists as well. But mostly these techniques and these characteristics of growth and these characteristics of cell division are referring to bacteria. So what I'm going to do first is I'm going to, in the first part of the lecture, I'm going to discuss various lab techniques that you would use to grow bacteria in the lab. You know that different bacteria, every living thing has a set of optimum conditions for survival. Every organism on Earth has a favorite temperature, a favorite moisture content, a favorite water content, a favorite pressure, favorite atmospheric pressure, and so on. And bacteria are no exception. So they will grow fastest and best at their optimum growth conditions. So what type of food do they like to eat? What type of pH do they like to live in? What type of pressure do they like to live in? Atmospheric pressure. What temperature do they live in? Do they require oxygen, or will they be killed by oxygen? So if you were growing a particular bacteria in order to study it, you would need to know these things so that you could keep your bacteria happy so that you can have enough of it to study. So there are many laboratories, many microbiology laboratories around the world that are studying just one particular type of bacteria, and they have to grow it up in large quantities so that they can do research on it. And so that's kind of what we're going to discuss in that section. You know, what are the available techniques for deliberately growing bacteria? And so we'll discuss those. And then in the second section, part B, we'll discuss what happens when you put a bacteria into a bottle full of liquid food. How fast will it divide? How fast will it eat up all the food and run out of food? And then if you want to keep it alive, if you're growing up a group of bacteria in a bottle or in a flask, as it's called a bacterial culture. It's called a bacterial culture. So if you're growing a bacterial culture in a flask, you want to know how much food do you have to put in. If you put in one bacterial cell, it'll divide, and then it'll divide again, and it'll double every few minutes. And then how long will it be before you have so many bacteria in that flask that they will have eaten up all the food? So how soon can you expect them to start dying? Is there a way that if you cool down the temperature, will they divide more slowly? That is usually the case. So that if you want the bacteria to divide more quickly, you can raise the temperature until you get to their optimum temperature, and then they will not divide any quicker if you go to a higher temperature. So that's why the temperature at which they divide at the maximum rate is usually considered to be the optimum temperature. If you lower the temperature below the optimum temperature, they will divide more slowly, and then you can keep a culture of them alive more for a longer period of time, rather. So there are a number of reasons why you might want to know what are the growth and cell division characteristics of bacteria, of a bacterial culture that you're growing in a bottle or in a large vessel that we call a bioreactor. We'll talk about that later. Okay, and then thirdly, in part C, we're going to learn the fact that not only is it handy to know what are the optimum conditions that a bacteria likes to live in, what temperature does it like, what food does it like to eat, what types of food does it need to eat in order to survive. It's nice to know all of those things if you're interested in growing up large quantities of bacteria. But what if somebody is infected with a bacteria and you don't know which bacteria it is, and you need to identify it so that you can figure out which type of antibiotic drugs that you want to give that person to cure them? One of the the most popular and traditional way to identify bacteria is to look at their unique growth and nutritional requirements. So, do they like oxygen or are they killed by oxygen? Right? So if you can figure that out, if you have an unknown bacteria that's infecting somebody and you can figure out that it's killed by oxygen, then you've eliminated a whole bunch of other bacteria that you know that this bacteria is not. And then if you say, well, is it gram-negative or gram-positive, you can figure that out, and that eliminates some other bacteria. And then you say, does it eat, what type of carbohydrates does it eat? What type of amino acids does it require? What temperature does it prefer? Does it need oxygen or not? If you can identify all of these things about the metabolism of an unknown bacteria, you can use the unique growth and metabolic requirements of a particular bacteria in order to identify it. And that's been the traditional way to identify bacteria that make us sick for about 100 years now. And then finally, at the end, we're just going to go through some specific techniques that you need to know in order to count bacteria. Okay, so if you have a culture of bacteria, it's nice to know how many bacterial cells per milliliter are growing in that culture, inside that food, inside that bottle full of food. How many bacteria do you have per milliliter? If somebody has a blood infection, you want to know how many bacterial cells do they have per milliliter of blood. That's called septicemia, by the way. Of course, that's called septicemia when you have bacteria that are living in your blood. And if you want to know how bad the septicemia is, you'd want to count how many bacteria per milliliter of blood that person has. And so we went over some of the techniques on how to count viruses. We learned how to determine the viral titer in the last lecture. And you do that with something called a plaque assay. And you look for cleared spots in a Petri plate, and you count the number of plaque-forming units per milliliter, PFU per mil. And then I mentioned when I was discussing that if you're doing bacteria, instead of plaque-forming units per milliliter, you want to count the number of colony-forming units per milliliter. So remember that if you have a Petri plate that has some food on it, like blood agar or LB agar or McConkey agar, if you put one individual cell onto that plate, it will stick to the plate in one place, and it will divide and divide and divide until it forms a lump of bacteria, a little tiny lump of bacteria that's called a colony. And if you plate a milliliter of blood or a milliliter of some bodily fluid onto a plate and you spread it around and then you count 20 colonies, that the next day you put it in an incubator and you count 20 colonies the next day, that means that you had 20 bacterial cells the day before. You had 20 bacterial cells to start with. And that's because you had 20 colonies, and therefore there were 20 colony forming units in that sample that you analyzed. Okay, so there are actually several different ways that you can count the number of bacteria per mil that you have in a solution, in a liquid, and we'll discuss some of those. So let's start in with the laboratory techniques about if we're deliberately trying to grow or cultivate a culture of bacteria, how would we go about doing that? What machines would we use? What techniques would we use? Okay, so if all you want to do is grow a specific type of bacteria, what techniques can we use to do that? Okay, so first of all, it helps if you know the nutritional requirements of your favorite bacteria that you're trying to grow. Also, if you know the temperature requirements, and also if you know the oxygen requirements. Does it breathe? Is it an anaerobe? Is it an aerobe? Is it a facultative anaerobe? Is it an obligate anaerobe? It's helpful to know those things. Okay, and then later on we're going to learn in the lab techniques how to count bacteria. And so you can count them directly using a special slide called a hemocytometer. Some of you have heard about this before, and I showed you this in one of the early introductory lectures. You can put them onto a plate, and then you can see how many colonies form, and that is you would count the number of colony forming units per milliliter. If you have too many colony forming units per milliliter in a solution, and you end up getting more than 300 colonies growing on a single plate, you can't really count that because it's statistically unreliable. If you have more than, for a standard size Petri plate, if you have more than 300 colonies on it, it's likely that you might have two you might have a colony that actually arose from two different cells, right? So if you have more than 300 colonies, you tend to underestimate the CFU per mil, because there's a very strong possibility that two cells landed on the same place in the plate, and then two cells gave rise to a single colony instead of two cells giving rise to two colonies. And so we tend not to count plates that have more than 300 colonies on them. By the same token, we don't count plates that have fewer than 30 generally. So you want to have, because those two would be statistically unreliable. The smaller sample size that you have, the less reliable it is statistically. That's just a universal truth. So you need to have as big a sample size as you can without it being so big that you might have had two colonies or two cells giving rise to a single colony. So generally, when you're counting bacteria colony forming units per mil, you want the number of colonies on your plates to be between 30 and 300. So let's say that you're plating, you put 1 mil of solution onto a plate and you smear it around, and then you look at the colonies that grow up. Let's say that if you put 1 mil on there and you get more than 300 colonies, you need to dilute it so that you get to a range where the plate does have between 30 and 300 colonies. So you do something called a serial dilution, which I explained in the last lecture, which is generally you assume before you even plate I keep saying plate, but it's a verb to microbiologists. You plate bacteria. That means you spread them on a plate. Okay, so that's called plating. Okay, so if you're plating bacteria, you're going to plate bacteria, you're going to assume that if you plate it, you will probably have too many colonies. And so you'll do a few dilutions before you even start. So you'll do a few dilutions and you'll plate each of those dilutions onto a plate. And then when you get some plates the next day from the incubator, you take the plates that have between 30 and 300 colonies and you multiply them by the dilution factor to get back to the original number. So the serial dilution was something that you did just to figure out how many colony forming units per mil there are, knowing that if there are more than 30 and you're plating a mil on each plate, knowing that if there are more than 300 rather, or fewer than 30, you can't use that information. So you do the dilutions ahead of time, expecting that you will probably have too numerous to count in the first plate. By the way, that's what the TNTC stands for, if you remember that from the last lecture. TNTC stands for too numerous to count. There's too many there to count. Okay, now there's another way that you can measure the number of bacteria that are present, and that is some of you may have taken chemistry class and you've used a machine called a spectrophotometer in the chemistry class. Spectrophotometer is where you put a test tube into this machine. The machine shines a light through the test tube, and then there's a receptor on the other side of the test tube that measures how much of the light actually got through the test tube. And therefore you can quantify or measure the amount of light that was actually absorbed by the liquid that was in that tube. Now, if you're talking about a color change or something, or if you're talking about chemicals or proteins in a tube, then you talk about how much light was absorbed, right, by the solution, by the chemical that was in the chemical or the solution that was in that test tube. If you're talking about bacterial cells, if you have a test tube full of cells, it's common practice not to call that absorption. The term absorption usually, or the term, well, it is actually absorption, but when you're talking about cloudiness of cells, the correct term to use is turbidity. Turbidity is a fancy word that means cloudiness, right? So if you look through a liquid and it's perfectly clear, then you say that that liquid is clear. But if you look into a test tube and it appears cloudy, that's because you have a lot of particles in there. In this case, the particles are bacterial cells, or they might be some other type of cell. And so you say that the solution is turbid. So turbidity is what you measure when you stick a test tube full of bacteria into a spectrophotometer, and then you measure the amount of light that is absorbed by the bacterial cells. That gives you a figure that you call the turbidity of the solution. So if so you can you can have an indirect measurement of the number of bacterial cells that are present by measuring the turbidity, by measuring the absorption of the bacterial culture in a spectrophotometer. Okay, so when you're growing up bacteria, they live in what we call growth medium. Growth medium means usually liquid, but sometimes solid food that they live in. So what they live on and what they eat is called their growth medium. The word medium is singular, and then the word media is plural. Right, so remember that there's a difference between medium and media. So if you have just one type, you would say that they grow in this particular growth medium. If you have two different types of bacteria that require two different types of growth media, then you would use the word media. All right, now beyond this, there are two types of medias. Basically, one type is called a complex medium, growth medium, versus defined growth media. Right, so complex media means, well, let me start with defined growth media first because it's easier to explain. Defined growth media means you know exactly what's in the growth medium. You had to put it together. You had to mix together a dozen different chemicals and amino acids and carbohydrates in an exact proportion. You had to add an exact concentration of amino acids, specific carbohydrates, specific vitamins, specific amino acids, in order to grow a very fastidious type of bacteria. Fastidious means it's very picky about what it eats, and it's very finicky. It's very fastidious about, it's very difficult to keep it alive because it's very picky and fussy about what it eats and how it's treated. So generally, defined growth media are used for specific bacteria that have fastidious nutrition requirements. Complex growth media is where you just throw a bunch of stuff together and there's an excess of everything. You don't know exactly what's in there because you haven't measured it all. You just throw a whole bunch of excess nutrients and foods, more than any particular bacteria would ever need, and anything will basically grow in that because it has everything that every bacteria would require to grow. So generally, when you're making a defined growth medium, you have to go to the chemical shelf and you have to weigh out 5 grams of cellulose and 2 grams of maltose and 3 grams of tryptophan and 5 grams of phosphate, monobasic phosphate, and then you mix them all together in a liter of water, and then there's only a specific bacteria that will grow in that. Whereas for complex growth media, what you do is you grow up a bunch of yeast, you grow up a bunch of Saccharomyces cerevisiae, you kill it, and then you grind it up so that everything that was in those yeast is now available and soluble, and you throw this yeast in with some water, along with an excess of the amino acid tryptophan, because for most bacteria and other living things, tryptophan is one of the limiting reagents. It's one of the things that you need the most of. And so you put all this, what's called yeast extract, into water, and then you throw in the bacteria, and it'll grow happily in there because the yeast produced everything that they needed to grow, and so then you killed them and ground them up and fed them to the bacteria, basically, and so the bacteria are growing on the components of dead yeast. You don't know there's 1,000,001 different components inside dead yeast, and you don't know exactly what they all are, and you don't know exactly how much of each is there, but you know that there's a lot more than the bacteria would ever need. So we call that complex media. Okay, so complex media, you just throw everything together that they could possibly want. This is generally non-selective medium. The best example of complex media is LB medium. Right, LB medium, you've learned this already, so you need to memorize it again. That LB medium is non-differential, it is non-selective. You see, a differential medium would be a complex or a defined growth medium. Sorry, a differential medium would generally be a defined growth medium that would allow one thing to grow but not something else, and that would be selective. So that would be a selective medium. On the other hand, if you have a medium that allows everything to grow, but it allows you to somehow differentiate between one bacteria and another, that's called a differential medium. And if it's neither differential nor selective, it's just a complex medium generally. So LB medium is a good example of a complex medium that is neither selective nor differential. Defined medium is where you add specific components to the mixture so that only certain things will grow. A good example of a defined medium is McConkey agar. So you know that McConkey agar contains crystal violet. Crystal violet interferes with the growth of gram-positive bacteria. So any bacteria that grow on a McConkey plate must have been, if they grew there, they must have been gram-negative. If they didn't grow there, they must have been gram-positive. We also add a pH indicator. We also add lactose to those plates so that, and a pH indicator, so that if the bacteria that did grow on the plate can ferment lactose, it will eat the lactose and then produce lactic acid as a waste product. Good example would be Lactobacillus acidophilus. So they would eat the lactose and then produce lactic acid as a waste product. The lactic acid would lower the pH and cause the pH indicator to turn pink. And that's why colonies that are bacterial colonies that are lactose fermenters appear pink on McConkey plates. Okay, now some of you gave a wrong answer on the exam, and it's perfectly understandable. Some people got it right, some people got it wrong. That's the purpose of having exams. Some of you said that if you had a gram-negative bacteria grow on the McConkey plate, and you can tell whether they ferment lactose or not because the colonies are either pink or gray. And then I ask you, what about this quadrant on the McConkey plate where nothing grows? And some of you said that proves that it's gram-positive and lactose non-fermenter. And I answered, my feedback usually was, how do you know that it's a lactose non-fermenter? Because it didn't grow. So you can't see whether it turned the pH into, you can't see whether it converted the lactose into lactic acid or not, because it didn't grow. So how do you know? And in fact, there are lots of gram-positive lactose fermenters, but they would not. There are lots of bacteria that can ferment lactose, but they're gram-positive, and therefore they wouldn't grow on the McConkey plate. So just because something doesn't grow on the McConkey plate doesn't tell you whether it's lactose positive or minus. It just tells you that it's gram-positive. Okay, so a good example of a complex medium where you just throw in everything would be LB medium. Good example of a defined medium would be McConkey plates, McConkey medium. Okay, so LB medium, the recipe for LB medium, I'll remember this as long as I live, because I used to make it every day, and it's one of those things that you want to make up a liter of LB medium, you memorize the recipe and you never forget it. Basically, you take 10 grams, I'm sorry, you take, I've almost forgotten it, but per liter, I'm just recalling this from memory, per liter you take 10 grams of yeast extract. It's a powder which contains all the amino acids and all the vitamins, because the yeast extract you got by growing up Saccharomyces cerevisiae and then killing it, grinding it up into a fine powder, and then selling that powder in a bottle, basically. So you put 10 grams of yeast extract, 5 grams of tryptone powder, which is tryptone powder is a mixed. It's a source of amino acids. You may not know this yet, but there's a protein in milk called casein. Casein, right? So there is a, where is it? There's a protein in milk called casein. If you digest it with protease enzymes, you end up breaking the casein down into amino acids. And so the tryptone powder is another rich source of amino acids, right? So to make up a liter of LB medium, you take a liter of water, you add 10 grams of yeast extract, 5 grams of tryptone powder, you adjust the pH to 7, usually with sodium hydroxide, and then you sterilize it. And that's it. That's all you need. Very simple. Takes you 5 minutes to do it. Okay, so sometimes if you're even lazy, you're too lazy to mix together the yeast extract and the tryptone and a little bit of sodium hydroxide. There are companies that for a few extra dollars will sell you the powder already mixed together. Right, so you don't even have to mix it yourself. And then when you've mixed it together, it looks kind of like this. It has like the flask on the right-hand side. It has kind of a yellow tint to it. It's a clear yellow color. Looks like apple juice or something like that. So that is definitely LB medium that you're looking at there. Okay, so then what you do is you take some bacteria and you seed or inoculate the medium right? You put in some bacteria as long as it's a pure little, you put in one bacterial cell. Usually you don't do that. You get an isolated colony on a plate. So that colony arose from one cell. So you take a metal loop and you take that colony off the plate and then you dunk it into this LB medium and then it will grow. It will grow in there. Right? So that's called inoculation, when you basically seed the flask that's full of medium with your bacteria. Right? Now, if you if you're growing it in a what's known as a liquid culture here, you have a liquid LB medium. What you've done is that's referred to as a broth culture when you grow the bacteria in it, right? As opposed to a plate culture. The difference between a broth culture and a plate culture is that the broth is the word for the liquid part of soup, isn't it? Right? So when you eat soup, the liquid part is called broth. And so when you're growing bacteria in liquid LB medium, we call that a broth culture. When you mix a polysaccharide called agar with the LB broth and then you heat it up, when it cools down, it will form a semi-solid that's kind of the consistency of jello. It's about as hard as jello, and we call that a plate culture. So here in the flask, which is up on the top right, we have LB broth, and you've seen pictures of LB plates. That would be LB; those would be LB agar plates. Right. So on the right, we have LB broth that has not been inoculated with bacteria yet. On the left, we have a broth culture where somebody inoculated it with some bacterial cells and then let it grow for a few hours, and notice that it's very turbid. So we mentioned that turbidity means cloudiness, and the reason why it's gone from being transparent yellow to cloudy yellow is because now we have billions of these bacterial particles in there. Yeah, so these are flasks, as you remember from your chemistry class. These type of bottles are called flasks, Flask, and these are flasks containing broth cultures. So that's on the left, that's a broth culture of a bacterium. Okay, now wait a minute. Did we miss a step? So we mixed the LB powder with water and then we threw the bacteria in there, but I think we missed something. There was already bacteria in that powder because you can't get rid of bacteria no matter what you do. So before you can inoculate your LB broth and then hopefully grow up a pure culture of your bacteria, you have to first sterilize the LB medium. So you have to sterilize the LB broth first. You have to kill off any bacteria that were already there so that you can inoculate that broth culture with your favorite bacteria and then ideally grow up a pure culture. You will grow up a pure culture of the bacteria that you're trying to grow. So how do we go about sterilizing the broth? Generally, we use a machine called an autoclave that uses high temperature and high pressure steam to kill every living thing that would be in there. Right? So an autoclave is one of the machines that a microbiologist uses every day. This is a large autoclave. So you can see here she's either opening or closing the door. The door is kind of like the door on a submarine or something like that. It's meant to withstand high pressure. You kind of turn this wheel and these metal bars come out and secure the door in place. And then you hit the go key, these buttons up here. These buttons are labeled 1, 2, 3, 4. Those are for what are called different cycles for the autoclave. So you put bottles full of LB broth or other things that you want to sterilize into the autoclave. You crank this handle so that the door is securely shut, and then you push one of these buttons and the boiler starts filling the inside with hot high pressure steam. And the temperature of the steam the temperature and pressure of the steam is high enough that after 20 minutes it will kill every living thing that could possibly be in there. And then you take it back out and you let it cool off and you have sterilized the LB. So now it's ready to be inoculated and create a pure culture of whatever bacteria that you want to grow. Okay, so just remember that this machine is called an autoclave. It's a way of sterilizing things with high pressure steam. This is a smaller autoclave than the one you're seeing there. So here you can put in these bottles. By the way, if you ever end up working in the lab, I'll save you some grief by telling you that you have to unscrew the lids a little bit so that they can breathe. Those bottles can breathe because as soon as the steam starts heating that liquid up, they'll burst. Those bottles will burst if the lid is screwed on tightly. Okay, so we talked about defined versus complex growth medium. Right, so this is an example of complex growth medium. You buy your own LB broth, right? So what do you know? I more or less got the recipe correct from memory. So you take 10 grams of, no, I didn't get it right. I mixed up, darn it, I mixed up the tryptone and the yeast extract. Okay, so it's been a few years since I've actually had to make this stuff. But yeah, so you put in 10 grams of tryptone powder, 5 grams of yeast extract, a little bit of salt, sodium chloride, 5 grams of salt, water, where it says water to 1 liter, that means you make it up to a volume of 1 liter, because what you do is you put the powders into a graduated cylinder and then you fill the cylinder up with water, because accounting for the fact that the powders take up some of the room. Okay, now there's a recipe for defined medium over here on the right, which is for this particular bacteria. You can see that it's got, sodium phosphate, 0.5 grams of sodium phosphate diabasic, 0.6 grams of sodium phosphate monobasic, zero 0.1 grams of magnesium sulfate, and so on and so forth. And these vitamins you have to add later after it cools off. You have to filter sterilize these vitamins because they'll be destroyed in the autoclave and so on. So complex versus defined medium. In the end, it all looks the same anyway, but in one case you're preparing a growth medium for a fastidious bacteria, and then hopefully nothing else will be able to grow on that complex medium, on that defined medium rather. All right, so I mentioned the fact that you can have liquid versus solid growth medium. Okay, so if you mix the components together and you add water, you have a broth culture, as we said. If you want to make a solid medium, so a broth culture, liquid medium, you just take those powders and you mix them with water and sterilize it in the autoclave. If you want to make a solid medium, you do all the same things, except you add about a gram of agar per liter. So agar is a polysaccharide that's originally derived from seaweed, and it's actually used as a thickener in some cooking. Like in some parts of the world, people use agar to thicken their soup, particularly in Asian cultures. In Chinese cooking, for instance, they sometimes use agar to thicken different types of food. I remember I worked in a laboratory one time and we ran out of agar, and the professor sent me to Chinatown downtown on Powell Street there's a large supermarket there where they specialize in Chinese foods, and he sent me there to buy like 10 kilograms of agar blocks that were meant to be used in cooking, and it's fine, it works, it's the same stuff. It's a lot less expensive if you buy it from a grocery store than if you buy it from a chemical supply laboratory. But it was an emergency and we needed some agar fast. Okay, so if you add about, I believe it's 1 gram of agar per liter in order to make solid medium agar plates, and then you pour them into these you heat it up. Agar comes as a powder. If you put it in water and then you heat it up, it will melt, and then when it cools off, it will dry into a solid. It will cool into a semi-solid that's about as strong as Jello is, for instance. And then you will have made Petri plates. So you heat up basically you mix together the stuff in a flask, and then you add the agar, you heat it in the autoclave, and then when it comes out of the autoclave, you pour a little bit of it into a whole bunch of these plastic plates that you buy, and you've made Petri plates. And that's what they look like. So you make up LB agar, and you make up the LB medium, you add a gram of agar, you heat it in the autoclave to sterilize it, and then as it's cooling, while it's still a liquid and hasn't solidified yet, you simply pour it into these Petri plates and then let them cool off, and they will solidify when they've cooled off. And so these are plates, that's a flask. There are other things that you can grow bacteria in, other types of medium, right? So you see there's a hand on the right-hand side of this image. In the middle, they're holding 3 test tubes. In the middle is a broth culture, so that's a tube that contains LB broth. And then on the tube on to the left of the LB broth is called an Agar deep tube. And what you do there is that tube is that is Agar. It's LB Agar. And then what you do there is you take some bacteria. It's for making something called a stab culture, STAB, stab. And what you do is you take a little bit of bacteria and you put it on the end of a sterilized wire, and then you stab the wire down to the bottom of the tube. And then if the bacteria is an aerobe, if it's an obligate aerobe, basically what you will have done is you will have carried a few bacteria all the way down the hole that you made when you stabbed the wire down to the bottom of the tube. And if the bacteria is an obligate aerobe, it will only grow at the top end of the tube. If it's an obligate anaerobe, it will only grow at the bottom of the tube. And if it's, a facultative anaerobe, it will grow all the way down. And so that agar deep tube, also known as a stab culture or a stab tube, is used to analyze the oxygen requirements of a bacteria, right? So you have an unknown bacteria, you want to know whether it's an aerobe, an anaerobe, or a facultative anaerobe or something like that. You put a little bit of it on the end of a wire and you stab it down to the bottom of that tube. You will have inoculated that solid medium all the way down, and where it grows in the tube will tell you about the oxygen requirements. Okay, now the tube over on the far right is called an agar slant or a slant agar tube. Notice that the agar is in there at a slant. So what somebody did was they melted some agar, they melted some LB agar, and they poured it into these tubes, and then they left it to cool off and therefore solidify at an angle. So you have this large surface that you can use to spread bacteria, and then generally what you do is you take a wire, a metal wire, and you get some bacteria on the end of it, and you kind of squiggle the bacteria around on the slanted surface, and then you stab it down into the bottom. And so the reason for that is if it's an aerobic bacteria, you'll get a lot of it growing on the surface because the surface part of that tube is larger than the bottom part, for instance. And then over on the far left, we have a standard agar plate. That's obviously an LB agar plate. You can see that it's not a McConkey plate, and you can see that it's not a blood agar plate. Okay, so that's called a stab tube, and that's called a slant tube. Okay, so here are some specialized agar mediums again. So on the left, we have a standard LB agar plate, which is neither differential nor selective. On the right, we have the McConkey plate, which is both differential and selective, right? It selects for gram-negative bacteria, and it allows you to differentiate between gram-negative bacteria that are lactose fermenters versus gram-negative bacteria that are lactose non-fermenters. And in the middle, we have a blood agar plate, which is where they take LB agar and they mix it with either sheep's or cow's blood, and then blood cells, giving it the red color. And then you plate things on there, and it is not selective because it allows both gram-positive and gram-negative and other things to grow on the plate, but it is differential because it allows you to differentiate between alpha, beta, and gamma hemolysis. Okay, so let's talk briefly about differential versus selective medium. Right, so LB agar, as I said, is neither differential nor selective because it doesn't stop anything from growing. And once things have grown on it, you can't really tell whether it's gram-positive or gram-negative or lactose fermenter or lactose non-fermenter or whether it's hemolytic or not. Okay, blood agar plates are differential but not selective. They allow anything to grow, whether it's gram-positive or gram-negative, but it does allow you to differentiate between alpha, beta, and gamma hemolytic bacteria based on whether it just completely destroys the blood cells or whether it partly destroys them or doesn't destroy them at all. Right, and the only confusing thing about this is that you would think alpha would be the strongest hemolysis, but it's not. Alpha is partial hemolysis. Beta is the strongest hemolysis, and then gamma is not hemolytic at all. All right, then McConkey agar is both differential and selective. It selects for gram-negative bacteria, and it allows you to differentiate between gram-negative fermenters versus gram-negative non-fermenters. Okay, and then the blood agar plate allows you to differentiate. It doesn't allow you to select for anything, but it allows you to differentiate for alpha, beta, and gamma hemolysis. So for example, Streptococcus mutans is alpha hemolytic, Staphylococcus aureus is beta hemolytic, and Klebsiella pneumonia is gamma hemolytic. It doesn't affect the blood cells at all. Okay, and then McConkey allows you to select for only the selection. You will only select the bacteria that are gram-negative, inhibiting the growth of gram-positive. And then if it's pink, it's a lactose fermenter. If it's not, if it's gray, it's not. Okay, so by the way, LB agar is also sometimes referred to as tryptone yeast agar. It's the same thing. Apologies for the different labeling, but tryptone yeast agar is also known as LB agar. And then blood agar. So tryptone yeast agar or LB agar is neither selective nor differential. Blood agar is differential but not selective, and McConkey agar is both differential and selective. Okay, so we already know about defined versus complex medium and differential versus selective. Okay, so we learned about the difference between a broth culture, which is grown in a flask or in tubes, test tubes, versus solid cultures that are grown on Petri plates or sometimes in stab tubes or slant tubes. If you have a group of bacteria that are all the same, growing in the same place, we call that a culture. So if you inoculate an LB flask with one bacterial cell, pretty soon you'll have a culture of that cell. Okay, we talked about the difference between defined and complex medium and selective versus differential medium. All right, there are two reasons why you might want to know a bacterium's nutritional requirements. Number one is if you're trying to grow a specific bacteria, there are laboratories in the world that only study tuberculosis, for instance, and they need to grow up tuberculosis, so it helps if you know what type of food it likes to eat and what type of oxygen requirements it wants, right? So if you're deliberately trying to grow a bacteria, that's one reason why it's necessary for you to know what its nutritional and physiological requirements are. On the other hand, maybe you don't care about growing it up, and in fact you're going to try and kill it, but you can actually use the specific nutritional requirements that every bacteria has in order to identify it. And so that's usually what a lot of these tests have been for. So for about probably a good 100 years, the main way of identifying a bacteria, if somebody has a bacterial infection, you want to find out which bacteria they're infected with so that you can give them a specific antibiotic that will kill it. You would identify it using the traditional methods, which are to analyze its nutritional requirements. So let's look at some of the requirements for cell growth, beginning with temperature requirements. Okay, so you already know that we call bacteria that like to live at moderate temperatures mesophiles. Mesophiles, the temperature for mesophiles is generally between 25 and 35. 37 degrees is human body temperature, but the bacteria that grow at that temperature are still considered to be mesophiles. That's a good tropical temperature, which is where humans originally evolved in the tropics. And so the humans are adapted to an optimum temperature of 37 degrees, and so are bacteria, a lot of bacteria that we classify as mesophiles. But there are some bacteria that like high temperatures, and we call them thermophiles. So bacteria that like high temperatures, like 70 degrees Celsius, would be classified as a thermophile. Bacteria that like low temperatures are called psychrophiles. 16 degrees or below would classify them as a psychrophile. Notice that all metabolism actually stops at 4 degrees, not 0. That's a mistake on my part. I apologize. So metabolism, particularly the metabolism and growth of bacteria and fungi and yeast, will stop at 4 degrees. Right. Now, that's why we keep our refrigerators at 4 degrees. If you go home and you look inside your refrigerator, you'll see that it's probably set between 4 and 6 degrees. In theory, bacteria and fungi and yeast that will spoil your food will not grow at 4 degrees. Okay, why don't we just freeze it? Why don't we take it to 0 degrees? Because freezing often destroys the texture of the food because it forms ice crystals inside the cells, right? So if you have meat or vegetables and you put them in the, or fish, you put them in the freezer, they, when you thaw them back out again, they feel kind of limp, and that's because the cells have been broken by the formation of ice crystals inside the cell. Right, so that's generally why you avoid freezing food unless you have to, unless you're keeping it for long-term storage. But if you're keeping food for short-term storage, it's moist food that bacteria or fungus will just love to eat, and bacteria and fungus would gladly spoil it if you left it out at room temperature. You put it at 4 degrees to stop the growth of bacteria and fungus and yeast. Now, generally, what happens is people are opening and closing the door of the refrigerator so much that the temperature is above 4 degrees. And then also, if the refrigerator is too crowded, it's generally going to be above 4 degrees. So most refrigerators, theoretically they should be set to 4 degrees, but if they're set to 4 degrees, usually the temperature is between 6 and 10 or something like that, depending on how much food is there and how often people open and close the door. And that's okay. So that's not going to stop the growth of bacteria, but it will slow it down. Putting the food at 10 degrees will slow down the growth of bacteria, but eventually, and yeast as well, and fungi, but it will eventually spoil. All right, if you have, okay, for most mesophiles, room temperature, most mesophiles grow at 37. That's their optimum condition. Room temperature is between 20 and 23, something like that. That's a little bit cold for them. So they will be happier if you put them into an incubator. So an incubator is a box that's heated to the optimum temperature that bacteria like. And so if you're trying to grow mesophiles, that happens to be 37. So here's a picture of an incubator. You can see the Petri plates on the top shelf and the flasks and the liquid cultures on the bottom. It has a transparent plastic door that you can look to see what's in there without having to open the door, because every time you open the door you let the heat out. If you look at the bottom, you'll see some red digits that say 37.1. That means it's 37 degrees Celsius rather. That's a C, I guess. And there's usually a thicker door in front of the plastic door, so that an incubator generally has two doors. There's a thick insulated outer door. You open it up, and then you get to look through the transparent plastic door. And then if you want to take the plates out, you can open the plastic door. Okay, this is another instrument. This is an incubator that's combined with a device called an orbital shaker, and this is for flasks. So you put the flasks in, you kind of snap them down. You can't really see in this picture because it's too small, but there's a wire ring around the bottom of these flasks that's holding each of these flasks in place, and you can turn on a wheel at the bottom that kind of shakes these tubes or shakes these flasks around in a circle. Why would you want to do that? You would want to do that if you want to aerate the bacteria that are growing in those flasks. And so we'll talk about orbital shakers in a minute. But if you're trying to grow a bacteria that likes lots of oxygen, you want to be able to stir the broth that they're growing in. You're certainly not going to stick a spoon in there and stir it the way you would soup, because the spoon is not sterile, so you would contaminate your broth culture. So the simplest way to stir it is simply to shake it. You have a machine that holds the flask and shakes it around in a circle, and that's called an orbital shaker, and it also at the same time keeps them at a certain temperature. Okay, here's yet another incubator. This one they're growing cell lines, human cells, in that particular incubator. Some laboratories are equipped with an incubator room where the whole room is heated up to 37 degrees, and you have, so those other incubators that I just showed you are relatively small. But some labs have a thing called an incubator room, and that again is for growing large quantities of mesophiles that have an optimum growth temperature of 37 Celsius. And what if we want to grow psychrophiles? Well, most laboratories also have something called a cold room. which is basically a big walk-in refrigerator. There are a lot of people that work in these rooms all day long. You have to wear your coat when you're inside because it's cold. It might be it might be between 10 and 16 or something like that. It's just cold enough to be unpleasant. It's usually very damp. It's kind of unpleasant to work in the cold room. If any of you are interested in going into biochemistry research, you'll probably spend a lot of your time in a cold room because proteins will break down and degrade more slowly at a lower temperature. So you do all kinds of experiments, column chromatography and SDS page, gel electrophoresis, and things like that. You do all of these experiments in the cold room. So you spend all day, it might be the middle of summer and it's hot outside, but you're stuck in this stupid damp cold room with your coat on. That is part of the life of a scientist. So this is a cold room. This thing here is actually it's a pen that's charting the temperature of the room. So every time somebody opens the door, the temperature usually goes up because the cold air goes out, and then they shut the door, the temperature goes down. And this pen is marking how many times somebody's opened and closed the door. This guy that's in here is a bit of an idiot because you're not supposed to leave the door open. You let all the cold out. So if this guy is standing in there with the door open, this pen is probably going up and recording a big bump on the graph, and then the professor is going to come and yell at him for leaving the cold room door open for too long and allowing the precious psychrophiles to heat up. Okay, so temperature, we have optimum growth temperatures for different types of bacteria. For mesophiles, it's 37 degrees. For psychrophiles, it's 16 or below. For thermophiles, well, if it's a thermophile, generally we're not going to be in the same room with it. At 70 degrees Celsius, humans, there are no incubator rooms that are at 70 degrees, and humans are in there because that's too hot for human survival. Humans are definitely mesophiles. Now let's move on to the requirement for oxygen. So we've already mentioned the fact that if you have an organism, including a bacteria, that needs oxygen to live, humans, for example, but lots of bacteria are what are known as obligate aerobes. So obligate means you have no choice. You are obliged to breathe oxygen. You need oxygen to live. Okay, so an example of a bacteria that needs oxygen to live, an obligate aerobic bacteria, is Bordetella pertussis. Right? I might ask that on an exam. Right? So give me an example of an obligate aerobic bacteria, Bordetella pertussis. That makes perfect sense because what does Bordetella pertussis cause, and where does it live when it infects humans? Right? It's a respiratory bacteria that is spread by droplet transmission by coughing, and it takes up residency in your lungs where there's lots of oxygen. Right? So if there are other places in your body where Bordetella pertussis could not live because there's no oxygen. Okay, if you have the ability to breathe oxygen, but you can convert to fermentation if you have to, you have the faculties to do anaerobic respiration if you must, then you are called a facultative anaerobe because you have the facilities, you have the faculties to grow without oxygen. Obviously, the best example of that is Saccharomyces cerevisiae, which is a yeast. But also, Staph aureus and Bacillus anthracis are both classified as facultative anaerobes. Okay, a microaerophile is a bacteria that needs oxygen, but it's killed by too much. Generally, the reason for that is because it has enzymes to do the Krebs cycle, to do glycolysis, the Krebs cycle, and the electron transport chain, but it does not have catalase enzymes. And so remember that if you're in the presence of oxygen, if oxygen mixes with water, you generate a little bit of hydrogen peroxide. Hydrogen peroxide is a very toxic, very dangerous free radical. And so many microorganisms and even humans have enzymes that convert hydrogen peroxide back into water and oxygen. And so if you breathe oxygen, you need oxygen, but you may or may not have those enzymes. If they're found in a bacteria, we call them the catalase enzymes. That's why we have the catalase test to be able to tell whether or not the bacteria that we're looking at actually makes catalase enzymes. So that's the catalase test. So that's generally what a one of the main differences between an obligate aerobe and a microaerophile is that an obligate aerobe has lots of catalase enzymes, so it can tolerate lots of oxygen, and in fact, the more oxygen, the better. Whereas a microaerophile does not have the catalase enzymes, but it still needs oxygen. But too much oxygen will kill it because it will end up generating too much hydrogen peroxide. So these organisms like to live in areas where there's a little bit of air but not much. Right, so an example of a microaerophile is Treponema pallidum. Remember, Treponema pallidum causes syphilis. Right. Okay, now what is an aerotolerant anaerobe? An aerotolerant anaerobe is a microbe that does not have the enzymes to do aerobic respiration. So it doesn't have enzymes to do the Krebs cycle or the electron transport chain, but it does have the catalase enzymes to detoxify hydrogen peroxide. So there's this kind of interesting relationship between microaerophiles and aerotolerant anaerobes. Microaerophiles have the enzymes needed to do aerobic respiration, but they don't have the catalase enzymes to detoxify the hydrogen peroxide. On the other hand, aerotolerant anaerobes do not have the enzymes to do aerobic respiration, but they do have the enzymes to detoxify the hydrogen peroxide. So it's kind of like if they could just exchange the stuff that they need, they would be perfect. They'd be facultative anaerobes, but they're not. Anyway, so they can use oxygen. They do not use oxygen. Aerotolerant anaerobes do not use oxygen, but they can tolerate small amounts of oxygen around. Example of an aerotolerant anaerobe is Streptococcus mutans, which you find growing on teeth. That's one of the causes of tooth decay. Ideally, your mouth is shut most of the time, so there's not too much air in there, but you speak and you breathe, and so there is some. Okay, so example of an aerotolerant anaerobe is Streptococcus mutans. All right, what about obligate anaerobes? Example, Clostridium botulinum is an obligate anaerobe. That means that they absolutely cannot tolerate living in an oxygen environment. So before long, oxygen will kill them eventually. Right? Now here's an interesting anecdote from history. You know that the Romans had fountains. If you look at Rome, the city is covered with fountains. And many of those fountains have been there for thousands of years because the Romans understood. They did not understand why, but they understood that if you pump the water through the air before you drink it, it's less likely to make you sick. Right? So if you pump the water up, if you have the water just traveling underground in pipes and people are drinking from the pipes, they will usually get sick if they drink that water. But if you pump the water up through the air into the sunlight and into the air, it is less likely to make you sick. So they used to have public watering holes where people in the town would come and they would come with a jug and they would collect the water from the fountain, and that water has been cleaned by aeration, by air and sunlight basically. So the Romans had no idea why that was. They had no idea what bacteria are. But now we know that a lot of the pathogenic bacteria are obligate anaerobes. And so if you pump the water up into the oxygenated air and up into the sunlight, you'll kill a lot of the pathogenic microbes that would normally make you sick. Now that's something that people forgot. As you know, the Romans knew that, but after during the Dark Ages, basically after the collapse of the Roman Empire, everyone around Europe forgot about the usefulness of building fountains, and they stopped. And so that's when we had in Western Europe and Northern Europe, we had lots of people getting cholera because they were no longer pumping their water through fountains, whereas fountains would probably help to correct that. So many pathogenic bacteria are obligate anaerobes, and pumping the water up into the air will kill those bacteria with oxygen. And the other thing is that the sunlight produces ultraviolet radiation, and when we get to the next lecture, you'll find out that bacteria are much more vulnerable to being killed by ultraviolet light than higher organisms like humans. So humans, if we have too much ultraviolet light, that will kill us. We'll get skin cancer or we'll get sunburn. But a little bit of ultraviolet light does not kill humans. But a little bit of ultraviolet light can, in fact, kill many bacteria. So that's why water can be sanitized, it can be cleaned, it can't be. We'll learn in the next lecture that there's a difference between the terms sanitation and sterilization. The word sterilization means that you've killed every living thing in that water. So if you want to sterilize water, the only thing you can do is use an autoclave. If you want to sanitize, that means you've killed a lot of them, but not all of them, right? So so a good way to sanitize water, a very simple, inexpensive way to sanitize water is to pump it through a fountain. So by extension, if you're stranded on a desert island someplace and you have a choice between drinking water from a fountain on the left or from a stagnant body of water on the right, you would drink it from the waterfall because that water has been exposed to air and sunlight and it's more sanitary than the water that's just sitting there in a pond. So the last water that you want to drink is stagnant. Stagnant means to stand in the same place. Right? So you don't want to drink still water that is not exposed to sunlight because that's probably filled with bacteria or protists or Naegleria fowleri or something like that. Whereas the water that's falling off of the water fountain has been exposed to the air and the sunlight and it's more likely to be clean. And as I said, ultraviolet light from the sun will also kill bacteria. So again, if you have a choice between drinking these waters, you want to drink the water that's been aerated, exposed to air, and been in the sunlight. Okay, now what if you want to if you're trying to grow a bacteria that is a known aerobe, right? Or maybe, so what is the best way to aerate a culture? The word aerate means to put air, to oxygenate, to get fresh air into the culture if you're growing a broth culture. Right? So if you want to grow a broth culture of a bacteria that you know is an aerobe, it grows fastest if it has air, then you want to aerate the culture. You want to aerate the broth culture. So in the laboratory, we have three machines, depending on how much aeration you want. We have three different machines that we can use to aerate a culture. And I'll introduce you to these three machines, and you do need to know what these three machines are. Okay, they are an orbital shaker or an orbital shaker platform, a tube rotator, and a rocker platform. Okay, so an orbital shaker platform is a moving platform where you put flasks onto the platform. There's a little spring that holds them in place, and then they basically rotate the bottom of the flask. It's basically, it shakes it around in a circular orbit called an orbit, that's why it's called an orbital shaker platform. And you can adjust the speed so that it shakes these flasks in a circular motion either more quickly or more slowly. If you turn up the speed, you'll shake them more vigorously, and then you'll get more air into just the act of swirling the broth around inside the flask will get air into the broth. A tube rotator is where you have this little thing that you stick test tubes in. The test tubes are filled with broth, and then you can adjust. It's a wheel, basically, that you stick test tubes in, and then you can adjust the speed that the wheel rotates. And then finally, there's a thing called a rocker platform, which doesn't rotate tubes and it doesn't shake flasks. You put a flask onto it, and it gently rocks it as if you were rocking a baby. Right. So obviously for obligate aerobes that if they want to have as much oxygen as you can give them, you can put them into an orbital shaker or a tube rotator. If you're dealing with a microaerophile, maybe the rocker platform is better because it will slowly rock the cultures back and forth, gently rock them, and it'll get a little bit of air in, but not as much as the orbital shaker or the tube rotator. Okay, so these are examples of orbital shakers. You can see that there's springs or little bars that hold the flasks in place. And then these knobs at the bottom, notice the one on the right, there are two knobs. One of them is controlling the temperature of those flasks, and the other one is controlling the speed, the orbits per minute that you're shaking them at. So you can shake them faster or more slowly, depending on how much aeration they want. So you can take the whole shaker platform and you put it into an incubator, And then you can, again, that's another way of keeping the temperature constant, so you can control the amount of temperature and the amount of aeration as well. There's a tube rotator. It's basically a wheel with holes in it that you stick you stick the test tubes in there, and it spins them around on a slant. You can speed it up or slow it down to spin the tubes faster or slower. Notice that the tubes are on a slant, which gives you a larger surface area that's in contact with the air. Right? So you see the surface area of this liquid is larger than it would be if the tubes were simply standing straight up. Now, these tubes have a breathing lid, right? So the lid does not actually fit airtight over the test tube, and you should be aware of that if you ever get a job in a laboratory as a technician on your first day. If you put a liquid into one of these tubes and put the cap on, and then you turn the tube upside down, the liquid is all going to come out because the tube is not the lid is not meant to be airtight. It's meant to allow air into this tube. So this is a breathing lid. Right, so you can see here that this lid snaps loosely onto the end of this tube so that when it's closed, air can still get into the tube from around the outside. So these plastic tubes with the breathable lids is exactly what you would grow a broth culture of bacteria in if you were just interested in growing a small volume of that bacteria. If you wanted to grow a large volume, of course, you would use a flask. Here's a rocker platform in the bottom here. You can see that there's some liquid cultures here that are just gently rocking back and forth, right? Like you were rocking a baby to sleep or something. So that would be for a microaerophile or something like that. Okay, what about obligate anaerobes? What if you don't want to aerate them? What do you do in that case? All right, there's several things that we can do to make an obligate anaerobe bacteria happy. One of them is called an anaerobic glove box. So when you're streaking the colonies onto plates and things like that, you want to do it in the absence of oxygen. So you have a little box that has a pair of gloves that reaches into the box, and you have an air pump so that you can pump the air out of the box, and then you can open up your bottles and you can spread them onto the plates in the absence of air. So that's called an anaerobic glove box. Right now, what about putting them in the incubator? Well, you could design an incubator. You could design and build an incubator that has no air in it, but it would be hard to get the air to stay out. It's simpler just basically to so you streak the bacteria onto your Petri plates in an anaerobic glove box, and then you put all of these Petri plates into a jar that's called an anaerobe jar, and then you pump the air out of the jar, and then you simply put the jar into a regular incubator. Right, so then you can have, in the same incubator, you can have aerobes and anaerobes growing side by side. It's just the only difference is that the anaerobes are in a jar that has no air in it. Right, so that's called an anaerobe jar. Okay, so how do you get the air out of the jar? You can pump it out. They're usually equipped with a pump, but there's another way, which is that you take a little package of chemicals, which is called an anaerobe pack, and you tear the package open, you drop it into the jar, and then you screw the lid shut, and these chemicals will react with the air, and they will absorb all of the oxygen. So there will be no oxygen in that jar. Okay, so anaerobe packs are somewhat expensive. Right, so if you end up working in a laboratory that doesn't have a lot of money to waste on luxuries like anaerobe packs, you can do a cheap alternative, which is to create something called a candle jar. Now it's actually quite clever when you find a good way to do something that saves money. That shows a high degree of cleverness. And what you do is you put your plates into a jar, and then you light a candle, and you put the candle into the jar, and then you screw the lid on. The candle will burn all of the oxygen out of the air, and then it will go out. Right? So what the candle will do is it will burn up all the oxygen and convert it into carbon dioxide. Okay, so the difference between an anaerobe jar with an anaerobe pack and a candle jar is that you cannot grow bacteria that are obligate anaerobes in a candle jar if they also don't happen to like carbon dioxide. Right? So if they don't like oxygen, but they don't mind a lot of carbon dioxide, you can use a candle jar. If they don't like oxygen and they also don't like carbon dioxide, you have no choice but to use the anaerobe pack to get rid of the oxygen. Okay, finally at the bottom, I mentioned that you can do anaerobic stab cultures. This is where you take the metal wire and you put some bacteria on the end of it and you poke it down into the bottom of an LB agar slant or an LB agar stab tube. So here's our anaerobic glove box. So what you do is you put your samples into this little box on the side, and then you shut the door, and then you pump the air out of this box, and then you reach in. There's a pair of gloves on the inside of this box. You put your hands into the gloves, and then you reach in and you take the sample out of this little side door, and then you can streak plate. You can streak plate or spread plate, spread the bacteria onto McConkey plates or LB plates or LB, whatever you like, or blood agar plates, whatever you like. And then you put them into an anaerobe jar and put in the chemical pack and screw the lid shut. And then you can let the air back into the glove box and take them back out and then put them in the incubator. Okay, so these are anaerobe jars. They're somewhat expensive. It's not trivial, the expense. And you can buy these little box of chemical packs, anaerobic packs that you tear one open and you throw it into the jar. You can see somebody's done that there with all these plates. And the chemical pack will suck all of the oxygen out. It will react it with some chemical crystals that are in that package. And then the oxygen will be gone and your bacteria can grow in the absence of oxygen. So here's an anaerobe jar where the air has been pumped out, and then somebody has also put in a gas, an anaerobe pack that contains the chemicals that absorb the oxygen. You can see these are blood agar plates that somebody's stacked up inside the anaerobe jar. And here's our cheap alternative to an anaerobe jar. This is a candle jar where you put some plates inside an old pickle jar, or ideally it should be a thick glass jar, because if you put a candle in there, it'll burn up the oxygen and substitute carbon dioxide. That generally takes up less room. So if you don't have a good strong jar, the jar may implode. It may implode, right? So the suction, it may create a suction that causes the jar to shatter inwards. So it should be a fairly strong jar, but you put a candle in there and you light it, and then the candle will burn up all the oxygen, convert it to carbon dioxide, and then the candle will go out, so you don't set fire to the laboratory, right? And so, as I said, the only difference between these two methods is that the candle jar generates a lot of carbon dioxide. So if you're trying to grow a bacteria that doesn't like oxygen or carbon dioxide, you can't use a candle jar. If you have a bacteria that just doesn't like oxygen, but it either likes or doesn't mind carbon dioxide, then yes, by all means use the candle jar. Now, this raises another term that I didn't mention before. There is a type of bacteria called a canophile, or canophilic bacteria. A canophile is a bacteria that does not like oxygen, but it doesn't mind carbon dioxide for the reasons that I just explained. And that type of bacteria can be grown in a candle jar. Right? So that is the definition. That's literally the definition of a canophile bacteria or a canophilic bacteria. Its characteristics are that it doesn't like oxygen, but it doesn't mind lots of carbon dioxide. Okay, so here we have on the left, the tube contains a broth culture, and on the right we have LB agar that's in a stab culture. Somebody has put a little bit of bacteria onto the end of a sterilized metal wire and stabbed it down to the bottom of that tube. And you can see that so it's a solid medium, it's LB agar in there. And so you can see that the bacteria is growing all the way down to the bottom, which means that it's probably a facultative anaerobe, which can grow equally well with or without oxygen. If it was an obligate aerobe, it would only be growing at the top. If it was an obligate anaerobe, it would only be growing at the bottom. Okay, so to summarize, right, so we talked about nutrition requirements, complex versus defined media, and so on. Then we talked about the temperature, incubators, different types of incubators, and we talked about aeration, different ways to either add air to a culture or to remove air from a culture. And when I say air, it's really the oxygen that we care about. So we're either adding lots of oxygen by aerating a culture, a broth culture, or we're removing oxygen by putting plates that have bacteria on them inside an anaerobic jar. All right, now let's move on to another subject, which is only slightly related to the previous one. So what about bacterial growth characteristics? Let's say that you inoculate a broth culture, how fast will you expect those bacteria to start dividing? And how much food do you have to put into the bacterial growth media to keep them happy? And how long will it be before they'll die because they run out of food? And will that depend on how much you aerate the culture? And will it depend on how fast you shake the culture? Will it depend on what type of medium you use? Did you use complex versus defined growth media? All of these things are classified as bacterial growth characteristics. And we really care about this because in this case, we're probably trying to grow bacteria for a specific purpose. We're trying to grow bacteria because we want to make lots of it and then do something with it, like do experiments with it. Or maybe we're trying to ferment beer. Right? So you take beer, you take barley and hops and you grind them up and you've got a fluid that contains barley and glucose and maltose, and then you throw in Saccharomyces cerevisiae. You say, how long is it going to be before the Saccharomyces cerevisiae divides and divides until they generate enough alcohol to choke themselves, right? So we discussed that earlier. Do you remember what the concentration of alcohol is at which point Saccharomyces cerevisiae dies? Do you remember that? I might ask it on a test. The amount is 12.5% ethanol. 12.5% ethanol is lethal to Saccharomyces cerevisiae, so that's why when you look at bottles of wine, it always says 12.5% alcohol. That's usually what it says. It doesn't usually say 20% or something like that, and that's the reason for that is because the wine was fermented until the Saccharomyces cerevisiae died, and it dies when it is it's choking on its own waste product, and that happens to be at a concentration of 12.5%. Okay, so there are a number of reasons. Let's say that we're producing insulin in bacteria. You remember we discussed that during the first lecture, that insulin is what diabetics inject themselves with. Insulin used to be extracted from the pancreases of pigs, right? We don't do that anymore. We simply took the human gene for insulin and put it into E. coli, into Escherichia coli, and then we have the Escherichia coli producing the insulin, right? So let's say you're a pharmaceutical company that's making insulin in E. coli. You need to know how much food you have to put into these big flasks that are growing the E. coli, and so you need to know what are the general characteristics of bacteria in a culture. How do they grow in a bacterial culture? And how does, if you plot the number of cells per mil that you have versus time, you will get something called a bacterial growth curve. And what does that look like? Okay, so this applies to, you would want to know about bacterial growth curves, how fast, when do they start dividing, and so on. How fast do they divide? Do they divide exponentially? The answer is yes, but not for the whole time. Anyway, so you'd want to know about bacterial growth curves. What are the characteristics of bacterial growth in a flask? What about this? This is what's known as a bioreactor or a fermenter. This is where you grow up large amounts of bacteria in this big container here. You can see how big that is because this is a staircase right here. So you can see that this is probably 2.5 meters tall, this container that's called a fermenter or a bioreactor. And the conditions inside the container are controlled by a programmable computer over here. And this is what we would call, if you're doing biotechnology and you're deliberately trying to grow a certain type of bacteria, or you're trying to grow a certain type of bacteria that's producing a recombinant protein that's been created by genetic engineering, this is what you would call a pilot scale fermenter. It's not big enough if industry is doing it, the industry usually works on a bigger scale. And if you're just doing little experiments in the lab, you would do it on a smaller scale. But if you're interested in kind of scaling up from the small to the large scale, you would do something, you would use what's known as a pilot scale, P-I-L-O-T, pilot scale fermentations. So this is not too big, not too small. This is just sort of like, we've learned everything we can learn from this bacteria from little 2-liter or 1-liter cultures in flasks. And now we're getting ready to move up to industrial scale. But first we're going to do fermentation on kind of a medium scale on a pilot level, on a pilot scale level, industrial pilot scale. So this is what I would classify as a pilot scale fermenter. And this large one here, which is about 3 stories tall or 2 stories, is a genuinely industrial scale, industrial size scale. So it ferments hundreds of liters of, it's used to grow hundreds of liters of bacteria for some reason. Somebody wants to grow them for some reason. Okay, so suppose you're deliberately growing bacteria for some purpose. You want to produce a recombinant vaccine, or you want to produce, you're brewing beer, any of those things. You need to know what the growth curve will be like. So what happens if you put one bacterial cell into some broth and then you allow it to grow under optimal conditions? Okay, so you create a whole bunch of LB broth and you put it into a flask or into a big bioreactor like that, and then you inoculate it with one cell. What's going to happen? How fast is it going to grow? Okay, so if you just do that, if you just take a whole bunch of growth medium and then you inoculate it and you let it grow, that's referred to as a batch culture, because when it runs out of food, you're not going to add any more food, or you're not going to take any of the bacteria out while the other and leave some of it in. Those are other types of fermentations that are called continuous flow and so on. But a batch culture is where you just have a big container full of growth media, you inoculate it, and then you measure how fast the bacteria grows. When you do that, that's called a batch culture. So what does the growth curve look like when you do a batch culture? And that's what I'm going to show you now. Okay, so generally, if you take a large container that's filled with growth medium and you inoculate it, this is where you put in 1 or 2 or 1,000 bacterial cells. Usually you grow a colony, you get an isolated colony from a Petri plate, and then you put that into the growth medium. That's called inoculation. You already know that. And then there's a short period of time that's referred to as the lag phase, which is where the bacteria are a little bit confused, quote unquote. They take a few minutes before they actually start eating. In fact, what they're doing is they went from conditions where they didn't have too much food to eat, and suddenly they find themselves in an environment where they have tons of food to eat. They have lots and lots of food to eat, but they have to make the enzymes that are needed to digest that food first. And so they take a few minutes to get ready to start eating. They take a few minutes to start producing the enzymes that are needed to start eating, basically. So that is called the lag phase. The lag phase occurs right after inoculation. Okay, now suddenly the bacteria are ready. They're ready to start eating, and they start dividing very quickly. And so they start multiplying and dividing exponentially. Right, so they, you know that E. coli at 37 degrees, E. coli and most other bacteria will divide every 20 minutes. And so that means every 20 minutes, the number of bacteria you have will double. It will double, right? And so if you double something every few minutes, that's called exponential growth. Okay, so after the lag phase, we have what's known as the log phase. And then eventually they run out of food, and as they're starting to run out of food in the batch culture, this is a batch culture we're talking about, as they start to run out of food, they will stop dividing, and they will just sort of sit there and basically just get fatter, right? They'll just sit there and get fatter, but they won't be dividing as fast. So the number of bacteria in the batch culture will not grow, will not increase. Right? Now, think about this and remember it for later, because the last lecture we're going to talk about is antibiotics and the mechanisms by which antibiotics kill things. And penicillin and all of the antibiotics that are related to penicillin will only kill gram-positive bacteria when they're dividing. So the penicillin interferes with the formation of the cell wall, but that's only a problem. Basically, the penicillin interferes with the construction of the cell wall, but a bacteria is only building a cell wall when it's dividing. Right? So when you have a mature gram-positive bacterial cell wall, it's already built the cell wall, and so penicillin doesn't hurt it anymore. Right? Penicillin will do the most damage to a gram-positive cell during the log phase, when the bacteria is rapidly dividing and every new cell has to build a new cell wall. Right? So that's interesting to know. That's important to know. That if you have a batch culture, if you have bacteria growing according to a standard growth curve, and you're trying to kill it with a penicillin or a penicillin-like antibiotic, you can only kill it it's only really vulnerable during the log phase, but not during the stationary phase. All right, and that's the reason why I just explained. It's because they are building, these cells are rapidly building cell walls when they're dividing, but after they've divided, they've built their cell wall, and so penicillin can no longer interfere with the construction of the peptidoglycan cell wall. Okay, eventually then they'll run out of food and they'll start to die, and so the number of living or viable bacteria in the batch culture will decrease. Right, so if you're growing up bacteria and you're trying to get to the maximum number of bacteria in that container, you would want to stop and harvest or collect the bacteria somewhere in between the log phase and the stationary phase. So here we have a graph. This is from your textbook, chapter 9. It's from the OpenStax textbook, right? So here we inoculate the bacteria here. And then we have that short period of time where the bacteria are kind of confused and they haven't yet created the enzymes they need to digest the food, and we call that the lag phase. And then they're all ready to start eating and reproducing, and so they start eating and dividing like crazy. They start doubling every few minutes. Whenever you double something every few minutes, it's increasing exponentially. Now, in order to see that, some of you that have studied math, if you know that if you have an exponential curve, it's a little bit easier to see what's going on if you plot it on a logarithmic scale instead of just a regular scale. So it is the exponential scale, but we call it is the exponential scale, but we sometimes call it the log phase because it's growing exponentially, and people plot the graph as they're watching it. People will plot the graph on logarithmic graph paper, or they'll just use a computer to convert the numbers into logarithms, right? So this graph that we're looking at here is actually a logarithmic graph. Okay, then the bacteria will stop dividing, then you get something called the stationary phase, and then you move into the death phase, or the period of decline, right? So these are the 4 phases of a batch culture. So remember, we're talking only about a batch culture here. And so somebody's growing up a batch culture, and then they're going to collect all the bacteria and break them open and get the insulin out and then purify the insulin. That's something that you would use for a batch culture. Right? All right, so for most purposes, if you're a bacteriologist, you're a microbiologist, and you're trying to grow up and isolate and purify a whole bunch of a specific bacteria, you would usually grow a batch culture, in which case the batch culture growth curve that I just showed you would apply. Right, so you'd be sitting there taking a sample out of your batch culture every few minutes and measuring how many colony forming units you have per mil, so that you can tell when you arrive at the stationary phase, you can stop and then collect all the bacteria, because there's no point in letting it continue because they're just going to die, right? So you want them to be growing, so you're counting and you're watching them grow during the log phase, the exponential phase. And then when they reach the stationary phase, you'll stop and you'll collect them, right? So you'd be measuring the amount of bacteria, the CFU per mil, as they're growing. Okay, now there's some other types of cultures that pharmaceutical companies do and scientists do for other reasons, right? So So one of them is called a continuous culture. Okay, so what happens is you have a flask that has bacteria growing in it, and you're constantly putting in new food. You know, they're eating up the food and they will exhaust the food. So what you do is you're constantly putting new food in as it's growing, and to prevent it from getting overgrown, you're continuously removing some of the bacteria, right? So you're adding new growth media to the top of the container, and you're simultaneously taking the broth culture that's filled with bacteria out of the bottom of the container at the same rate, so that the volume in the container stays the same, but you're constantly removing some of the culture and then doing whatever you want with it, right? That's called a continuous culture or a continuous flow culture, where you keep a culture going indefinitely and you're removing some of it from one end and you're adding extra food, new food to the other end. All right, now there's a variation on a continuous culture, something, or it's actually a variation on the batch culture, and it's called a fed batch culture. Right? So what you do here is you don't take any bacteria out, but when the bacteria start to exhaust the food, you just add more food, right? So you add concentrated food, basically. So you make up some LB broth that is 10 times more concentrated than it would normally be, and you add a little bit of that to the container as the bacteria start to run out of food and they start to go into the stationary phase. You can keep them going in the log phase and the exponential phase by adding a little bit of concentrated food. So that's called a fed batch culture. Okay, and then a continuous culture, as I explained, you're adding fresh food to one side while you remove an equivalent amount of the culture, cell-containing culture, from the other side. Right, so this is a diagram of a continuous flow culture. Right, so here we have a propeller that's aerating the culture. That when you have a propeller on the outside of a ship, you call it a propeller. When you have a propeller inside a bioreactor or a fermenter, you call it an impeller. Impeller, impeller. Right, so there's an impeller here which is stirring it to keep it aerated, and at the top we're adding new food. And at the bottom, we're removing an equivalent volume of the bacterial cells and then isolating the insulin out of them or whatever you like. So this is a diagram of a continuous flow culture. Basically, the purpose of this is to keep the culture going in the lag phase, in the exponential growth phase, indefinitely. Right? So you keep them growing in the exponential growth phase indefinitely. Okay, so a bioreactor or a fermenter can be of several sizes. This is a little tiny one that holds 1 liter. This is a large one. So again, you can see the staircase over here, so you can judge how big those bioreactors are. All right, so to summarize, we talked about a batch culture and the characteristics of growth that are associated with when you're growing bacteria in a batch, just a bunch of it. And you're not going to add any food. You're just going to take a bunch of liquid medium and inoculate it and then let it grow, and you're probably going to collect the bacterial cells or harvest the bacterial cells when they just reach the stationary phase. That is in contrast to a continuous flow culture or a fed batch culture. Fed batch culture, you're not taking anything out, but you're adding concentrated food to keep it going in the in the exponential phase a little bit longer. Continuous phase, you keep it going in the log phase continuously. Right. And so for a batch culture under ideal conditions, you can characterize it with these phases. The length of each of those time periods, the length of the lag phase versus the length of the log phase and the length of the stationary phase, those things are usually unique to each type of bacteria. So the length of the lag phase would be different for E. coli than it is for Streptococcus aureus or something like that. All right, just for fun, I've done this exercise before, but just to demonstrate the amazing utility of having bacteria doubling every 20 minutes, suppose this principle applies equally well to fermentation as it does to, when you're deliberately trying to grow bacteria. It equally applies to food spoilage. Right? So, and making people sick. If you run a restaurant, you need to know about this kind of thing as well. So suppose you make a custard pie. A custard pie is basically just made out of eggs and sugar, so that's a lot of protein and a lot of sucrose, the two things that bacteria like to eat most. And suppose that somebody cuts the pie with a dirty knife and they inoculate the pie with 200 cells at 8 a.m. Right? So let's say that somebody made this custard pie for a picnic at the beach. Somebody cut it with a dirty knife and inoculated it with 200 cells at 8 a.m. It sits in the hot sunlight at 37 degrees. Let's assume that it's in the shade so the ultraviolet light is not killing the Salmonella typhi. And so it's in the shade, but it's a high temperature. It's a hot day, right? It's a picnic, and it's 37 degrees. You leave it out for 12 hours, and you have 20-minute generation times. Okay, so if you cut that pie with a dirty knife at 8 a.m., at 8 p.m., you started out with 200 cells at 8 a.m. How many Salmonella typhi cells will be in that pie by 8 p.m. if it stays at 37 degrees in the heat all day? Let's figure that out. Okay, so that there's 12 hours. 20 minutes, 20 minutes occurs 3 times every hour, right? So 12 times 3 is 36. That means we have, in 12 hours, we have 36 doubling times. So we take 200 cells and we multiply it by 2 raised to the exponent 36. So you multiply 200 times 2 raised to the 36th power. That's a lot of cells, isn't it? Right? So you started out in the morning with 200 cells. By 8 o'clock at night, you have 10 trillion Salmonella typhi cells in that pie, right? Which is why for many types of food, if you have many types of food in restaurants and things like that, you're supposed to throw the food away after it's been sitting under a hot counter all day. In fact, you're supposed to throw it away much more often than every 12 hours. You're supposed to throw it away every couple of hours to avoid bacterial growth, making people sick. Right, so that is why if you run restaurants and pastry shops and things like that, you keep the produce cool, but if it's in a hot area, if it's under hot lights or something, you have to throw it away regularly because the doubling times for pathogenic, the doubling times for most pathogenic bacteria are about 20 minutes at a high temperature like 37. Doubling time is probably about 35 minutes at room temperature, so it slowed down a little bit, but it's still a dangerous growth rate. All right, now let's talk about analyzing the nutritional and temperature requirements and oxygen requirements of a bacteria in order to identify it. So we said that this is the traditional, this is for 100 years, this has been the traditional way to identify bacteria by looking at what types of things they can grow on, what types of food, do they require oxygen, yes or no, and so on. Okay, so first of all, you generally what they used to do in hospitals and what they still do in many hospitals around the world, but not all of them, is if somebody's infected with a bacteria and you don't know what they're infected with, you take a sample of their blood, for instance. You divide the sample of blood into two aliquots, and then you streak some of it onto a blood agar plate. You streak some of it onto two blood agar plates, and you put one of the blood agar plates into an aerobic incubator and the other one into an anaerobe jar. And then you come back the next day and you look to see, did it grow in both? If it grew in both, then it's obviously it's a facultative anaerobe. If it only grew in the anaerobe jar, then it's probably an obligate anaerobe. If it grew only in the aerobic incubator, it's probably an obligate aerobe. Right? So you can eliminate some possibilities that way. And then you look at the blood agar plates and you ask whether it did alpha beta gamma hemolysis. You would have also streaked two samples of it onto McConkey plates and put one of the McConkey plates in the incubator, the aerated incubator, and the other one into the anaerobe jar. And then you look at that as well. So then you can tell whether it's a gram negative or gram positive, whether it is whether it's a lactose fermenter, yes or no. That's a common question for when you're doing a coliform count to assess the drinkability of water, for instance, or the drinkability of milk. You do a coliform count. And finally, typically what you would do is then you would take of these colonies that have grown on the McConkey plate or the LB Agar plate, you would take a colony and you would smear it onto a slide and then you would stain it, and then you would be able to see, you would look at it directly under a microscope and you'd be able to see whether it's gram-negative or gram-positive, and then furthermore you'd be able to see the cell morphology. So does it have flagella, yes or no? What are the arrangements of the flagella? Is it a coccus or is it a bacillus, or is it a vibrio or is it a spirochete? You would do all of these things. So looking at the morphology combined with the physiology, meaning does it breathe oxygen or not? Does it ferment lactose, yes or no? So you combine an analysis of the morphology with an analysis of the physiology, and that's how you identify the bacteria. That typically takes about 2 days, 1 day, 2 days. So you get a patient and you do the blood sample, you streak the plates and you put them in the incubators, and you come back the next day and you read the results and you do the smear and you look at down the microscope. That's about 24 hours to do that, to identify the bacteria. Your patient might be dead by then, right? So that's a long time. So alternatively, these days, PCR, the polymerase chain reaction, is starting to take the place of these traditional methods of analyzing the morphology and the physiology of the bacteria. So these days what they do is they take somebody's blood and they directly put it into the PCR machine and they do a polymerase chain reaction to see which genes are present. So does this person's blood contain genes from Salmonella typhi, or does their blood contain genes from Staph aureus? And that's how you would identify the pathogen. But these techniques of identifying using morphology and physiology are still in use in many hospitals around the world, so we are expected to know them. And so you make 2 plates, you put one of them in the anaerobe jar and the other one in the incubator. You do the same thing with 2 McConkey plates. Right? You can do a stab culture as well the first day, and then you can tell. Now these stab tubes are very informative here. So I want you to pay close attention to these stab tubes. Okay, so notice on the far left, if you do a stab down all the way through this LB medium, solid medium, right? So you have if it's an obligate aerobe, it will only grow in the top. Right? If it's an obligate anaerobe, it will only grow in the bottom. Typically, a facultative anaerobe grows mostly in the top, but it grows a little bit all the way to the bottom. Right? So the growth is, for a facultative anaerobe, the growth is not even all the way down to the bottom. Right? Now, the aerotolerant anaerobe generally does grow all the way through with the same density, and a microaerophile will grow to great density just below the surface because it doesn't like too much oxygen. So it's not going to grow up here where the air is, and it's not going to grow down here where there's no air, but the microaerophile will grow very well where there's a limited amount of oxygen. Right? So look at these growth patterns and analyze them in the context of obligate aerobes, obligate anaerobes, facultative anaerobes, aerotolerant anaerobes, and microaerophiles. Yes, that would make a good exam question. So furthermore, you can look at what types of things the bacteria likes to eat, but what types of waste products does it produce is also a question that microbiologists often ask. And so will they ferment certain sugars? Will they turn those sugars into acids, right? So you might have a pH indicator in the stab culture in order to check for the production of acids. And also you can check to see if a bacteria produces gas. So we learned already that well, let me ask you, can you tell me an example of you know the answer, can you tell me the name of a bacteria that's an obligate anaerobe which generates gas when it eats? Right? So it has to be in an environment that has no air, and when it eats its food, it generates gas. And it does things like, for example, causing cans to swell up. right? So you know that which one does that, right? Clostridium botulinum. So you know that Clostridium botulinum is an obligate anaerobe, and you know that if it gets into a can of food, it's growing in there, it will cause the can to swell up because it's producing gases as it consumes the proteins that are inside the can. So these are examples. This is another way that you would examine that. This is another way that you would assay that. So the slant culture, the slant tube on the far right is demonstrating that the bacteria that was inoculated into this tube generates acid. It's a lactose fermenter. The tube that's on the far left is an obligate anaerobe because it only grew at the bottom of the tube, and it generated gas, which caused it generated this bubble here at the bottom of the tube, so it caused the LB agar to kind of be pushed up into the tube. Okay, so these are some of the selective and differential media that we would use to make these kind of analyses. Right, so you already know about the catalase plus minus test. You know about the coagulase test as well. Okay, now there's so many of these tests that it's standard protocol to use for technicians in the hospital who are analyzing bacteria. They use something called a dichotomous key. Right? So the word dichotomy means a choice of two. It's either this or it's that. If it's not this, it must be that. Right? You might have heard the expression a false dichotomy. You know, when somebody says, "Are you with me or are you with them?" Right? And then you say, I'm not with either of you. That's a false dichotomy, right? So a dichotomy is a binary choice of two, right? So people who design these tests have come up with things called a dichotomous key, which is basically a flow chart which tells you which tests to do in which order in order to give you the answer, in order to definitively identify a bacteria. Right, so a dichotomous key is a flow chart that tells you which tests to do in which order. Here's an example of a dichotomous key, right? So you take your bacteria and you stain it if it's gram positive or negative. If it's gram negative, you proceed this way, and then you do a test to see if it ferments glucose. If it ferments glucose, does it produce acid or does it produce acid and gas? If it produces just acid, then it's this one. Right? If it produces acid and gas, then you do another test to see if it's mobile or motile at 37 degrees. If the answer is yes, then it's that one. If the answer is no, then you check another test to see if urea is hydrolyzed, indole is produced. And then at the end, finally, you can get down to the bottom here, and you can definitively identify a bacteria if you do all of these tests in this order. So I don't expect you to memorize these tests. I just want you to know what a dichotomous key is, and I would expect you to know how to use a dichotomous key if I gave one to you. Right? So I would list a whole bunch of experiments, and then I would tell you the result, and I would show you the dichotomous key, and I'd say which bacteria is this. All right, now there's another thing that you can use basically to do a dozen different physiological tests at once. And it is a very expensive test that comes as a kit, and it's called an Enterotube 2. As you can tell from the name, it was meant to identify enteric bacteria that live in the gut. Right? So it's an expensive test that's used to identify a bacteria in one day. It contains about a dozen different tests, and I'll show you what it looks like. Okay, so you buy this thing called an Enterotube, which is quite expensive. It has a little metal loop on one end, and then there's a tube that contains a whole bunch of individual compartments. And then what you do is you dip let's say you have a blood sample or a urine sample or a stool sample. You dip the loop in that sample, and then you pull the loop from the other side of the tube. You pull the loop so that it drags the bacteria through all of these compartments. All of these compartments have generally pH indicators or other chemical indicators that give you a plus or minus result. And then the next day you come back and you read all the plus or minus results in the tube, and then you enter a number into the computer which tells you exactly which bacteria it is. So this is an Enterotube 2, right? And so underneath this end, if you take that cap off, there's a loop in there. You just dip that loop into the blood sample or the urine sample, and then you take off the other end, and then you pull the wire from this end, and that will pull the loop which contains the bacteria through all of these compartments, and then it'll inoculate all of these compartments, and then this chart tells you how to score the color change the next day as a plus or a minus, and then you enter that number in a computer, and it tells you exactly which bacteria it is. Right? So you don't need to know how to use an Enterotube, you just need to know what it is. Okay, so here's somebody pulling the wire through from one end to the other, right? And then you read all these results, and then you look at a chart, you get a number from reading the plus or minuses in these different compartments, right? So that's an expensive way of identifying a particular bacteria using metabolism. So here, this particular pattern at the top, that particular pattern of color changes identifies the bacteria as Proteus vulgaris. The one below that is another one. The 1 second from the bottom, that particular pattern identifies the bacteria as E. coli, and so on. All right, let's talk briefly about serology. The word serology refers to the word serum. The word serum means the liquid portion of your blood. And the serum is where you would find antibodies. So whenever you hear the word serology, it is usually referring to tests that involve antibodies. It's usually referring to tests that involve antibodies. Okay, so one method that you can use to identify bacteria using serology is you take a blood sample and then you look using various methods to see which antibodies are present in a person's blood, and then you know which bacteria their immune system has been exposed to. You can read those results in a few hours, basically, less than a day. There are some other types of tests that you can do that are also classified as serology, and this is where that you take, instead of having your body make an antibody that binds to a particular bacteria, you buy antibodies that will attach to certain bacteria from a commercial supplier, and then you put those antibodies in the bottom of a small plate called an ELISA plate, which we'll talk about in a second. And those antibodies that are stuck in the bottom of the ELISA plate will actually grab hold of the bacteria if it happens to be there, and then you just wash out the plate. The bacteria that the antibodies were meant to hold on to, that will still be there, and then you can identify that that bacteria is there. And that is something called an ELISA test. We're not really going to go into the detail of an ELISA test. That would be a topic for a more advanced course, right? But that's another, the ELISA test is also a serology test of sorts. And finally, you already know about the Lancefield grouping test, right? So the Lancefield grouping test, Lancefield group A, Lancefield group D, and so on, is another way of identifying. You take the bacteria and you put it onto a bunch of little circles in a card that you buy. You buy the entire kit. You buy the entire test as a kit that comes with about a dozen little bottles. Those different bottles contain different types of antibodies, and then you mix these antibodies with the bacteria, and then you look to see if which antibodies cause the bacterial sample to agglutinate. In other words, to form clumps, and that's how you group all of the bacteria according to the Lancefield test. So the ELISA test and the Lancefield tests are both two examples of serology tests that are used to identify bacteria. The Enterotube 2 is not a serology test, obviously. PCR is not a serology test because you're not using antibodies necessarily. You're looking for the presence of genes from specific bacteria. All right, let's talk briefly about the ELISA test. I don't really expect you to know all the details. I expect you to know some of the details. All right, so ELISA stands for Enzyme-Linked Immunosorbent Test. So what you do is you take specific antibodies and you put them at the bottom of something called a microtiter plate. Right, a microtiter plate is a little tiny plastic plate that has 96 small holes in it. Those holes are called wells. Right, so at the bottom of each of these wells, you'll put specific antibodies, and then you'll load samples into each of these 96 wells, and then if that sample contains a bacteria that antibody will attach to, the bacteria will end up getting stuck at the bottom of that particular well. And then you analyze whether there's a bacteria there or not using an enzyme. So this is use the antibody to capture different bacteria, and then you use an enzyme to identify that the bacteria is there. You typically read the result in a machine called a microtiter plate reader. Right? So you need to know the word microtiter plate reader. It is also referred to as an ELISA plate reader. But the problem with the term ELISA plate reader is that we've now evolved beyond just using microtiter plates to do ELISA tests. We now use microplates for all kinds of tests. So they had to change the name from ELISA plates to microtiter plates, and then they changed the name from an ELISA plate reader to a microtiter plate reader. Okay, so here we have an ELISA plate or a microtiter plate. Right, so notice that there are 96 wells. We have there are 8 rows. Sorry, 8 column. I guess that's 8 rows and 12 columns, right? So 1 through 12 going this way, 1 through 8 going that way. Total is 96. These little holes are basically just little containers. You put either enzymes or antibodies at the bottom, and then you load your samples into it. Right. So then you capture the bacteria with an antibody, either a protein or some other type of antigen, and then you add another antibody that's conjugated or attached to an enzyme that tells you that it's there, right? So the enzyme will react and cause a color change, and then you know that the bacteria or the protein that you were looking for has actually been captured there. You put the whole thing into a microplate reader that gives you all 96 results at once. So this is a mass production way of doing these types of experiments. As I said, the polymerase chain reaction is a machine that amplifies specific genes. We're not going to go into the details of how the polymerase chain reaction works for this course. We discuss how PCR works in Biology 200 and also in Biology 234, but we're not going to bother with it here. Suffice it to say that it's a way of amplifying genes so that you can tell that genes that are present in very small numbers, you can actually use it to detect genes that are present in very small numbers because the PCR machine makes multiple copies of them. Okay, so you take a sample, you put it in a PCR machine, which is referred to as a PCR thermal cycler. Right, so you put it in a PCR thermal cycler. You include 2 things that are called PCR primers. A primer is basically you're putting in DNA polymerase. DNA polymerase will extend from a primer. So you have to have 2 primers that correspond to the gene sequence that you're looking for. So you call those PCR primers. You're looking for particular genes that are unique to one type of bacteria. We refer to those, by the way, as barcode genes. Right? So barcode. What is a barcode? A barcode is what's on packaging so that you can use a laser scanner to identify it when you go to the supermarket. So you go to the supermarket and you buy your food. They run a laser over the barcode, and that gets entered into the cash register. So the barcode at the supermarket, the barcode is a way of identifying what the product is. Okay, somebody who invented this technique had a sense of humor, and they're using the same terminology to refer to the fact that there are 1 or 2 genes that are unique to E. coli, and there are 1 or 2 genes that are unique to Staph aureus, and so on. And so if you put in PCR primers that correspond to that unique E. coli gene, that gene will be amplified, and we can treat that gene, if it's there, as a kind of a barcode that is used to identify the presence of E. coli. Right, so we call those barcode genes, right? So you should know that term. A barcode gene is a unique gene that's unique to a specific organism. Doesn't have to be bacteria, any organism on Earth. There are a few dozen barcode genes that are unique to humans, and so you can tell if you have some tissue, you can tell whether that's human tissue or fish tissue, and if it's fish tissue, you can tell what type of fish it is based on the barcode genes that are present. A friend of mine, if any of you go to UBC, you might take a higher level biology course from a woman named Pamela Callis, who's a professor at UBC, who's an old friend of mine, and she was telling me that they have a course at UBC where they use PCR machines and barcode genes to identify different things. And she sent some of her students into in the student union building at UBC. There's a sushi restaurant that says we make our sushi with 100% salmon, BC salmon. Well, you can identify BC salmon using a barcode gene. And so she sent her students to that restaurant to get sushi, and they brought it back and they did the barcode test on the fish, and they found out that it was only about 20% salmon and it was 80% Alaskan pollock or something like that. So that was very embarrassing, but that's exactly the kind of thing that you can use barcode genes for. When you go to the restaurant or you go to a fast food place and they say this hamburger is made with 100% beef, you can take some of it and you can run it in the PCR machine and you can tell that that's not 100% beef, that's 50% beef and 50% horse meat or something like that, which happens a lot of the time. Anyway, so barcode genes can be amplified using a PCR machine, and you get the results in about 2 hours. And so, as I said, the traditional method for identifying bacteria was based on morphology and physiology. So you look at the shape and the type of the cell and the arrangement of flagella. You look whether it's an aerobe or a facultative anaerobe or whatever. And then you have the answer in a day or so. But that method is becoming obsolete, and it's giving way to PCR as the main way of identifying contaminants. So PCR will give you the answer in 2 hours or less. They have some new PCR machines that'll amplify barcode genes in 20 minutes or so. When I was a graduate student, I was in my 20s and 30s. The PCR machines were just beginning to be a thing. They had just been invented in the late 1980s, and so in the 90s, when I was doing graduate studies in the early 90s, they had just become a common thing to have in the laboratory, and they were huge. They were about 1 meter long and half a meter wide and half a meter tall. They were big, and they were cooled off by water. Now they're pocket-sized. Right? So people can go out into the jungle and they can take a sample of water out of the jungle pond, and they can analyze it in a PCR machine that they brought in their pocket. So the technology for polymerase chain reaction and using it to identify things, including bacteria, is rapidly advancing, and it's very convenient, very rapid, and very cheap at this point. So here's a laboratory benchtop PCR machine. You can see how big that is. You can see how big the buttons are. Those are meant for human fingers to operate. And this is a PCR field kit, right? So you take this little pocket-sized PCR machine, and you can go out and you can analyze the forest floor and see if that's septica on the forest floor, or is this pond contaminated with Naegleria fowleri or something like that. So PCR, 2 hours or less. The technology used to be expensive, but now it's cheap and it's been miniaturized. And so I have a feeling that in a few years, in the developed countries that have lots of money to spend on their hospitals, PCR will completely take the place of morphological identification of bacteria in a few years. Okay, let's finish off the lecture by discussing various ways to count bacteria. How do you know how many bacterial cells you have? Okay, so for example, when we were doing our batch curve growth characteristics, somebody had to count, when they were measuring the lag phase versus the log phase versus the stationary phase. The only way that they could plot a curve like that is if somebody every 5 minutes took a sample of the batch culture and counted the number of bacteria that were there. Right? So there are many reasons why we need to quantify or to count how many bacteria are in a sample. So there are 3 ways to do this. One of them is direct, and it's a direct count in a hemocytometer. Some of you have heard of a hemocytometer before. It's a slide. It's a microscope slide that has a grid etched into it that you put your bacteria onto the grid, and then you literally count it. The grid makes it easier to keep track of the number without losing your place, right? So it's a gridded microscope slide that you use to count the bacteria directly. Okay, the next method is you can take the bacteria, you can take a sample, a liquid sample that has bacteria in it, and you can plate 1 milliliter of it onto a Petri plate, and then you put it in the incubator. You come back the next day and you count the number of colonies, and you assume that every colony was generated by an individual bacterial cell in the beginning. If you're counting colonies between 30 and 300, that is probably a fair assumption. If there's more than 300 colonies, you can't conclude that every colony was caused by one bacterial cell. Two might have given rise to one colony. But that is called, when you do that, that's called the CFU per mil, or colony forming units per mil. All right. The third method is an indirect method, which is to measure the turbidity. So you take a test tube full of bacteria and you stick it in a spectrophotometer and you measure the amount of light that's absorbed if the light is 600 nanometers, right? So we call that the A600, right? So the A600 is the amount of light that's absorbed, the amount of 600 nanometer light, 600 nanometer wavelength light that is absorbed by a bacterial culture. So we call that the A600. It's a measurement of turbidity. Right, now, here's an interesting thing. What is the difference between the bacteria that you count using the hemocytometer, colony forming units, and turbidity? The difference is that counting the CFU per mil by plating the bacteria will tell you the number of viable bacterial cells, right? So let's imagine, just think back to when we did the batch culture growth curve. What did that curve look like when it went from the stationary phase into the death phase? The curve started to slope down because the bacteria started to die. Right now, that means that whoever drew that graph was counting the live cells, the viable cells, the cells that would still grow, right? They were not counting the dead cells. Because in fact, if they counted both live and dead cells, then the line would have just stayed level. The line would not have dropped because the same number of bacteria would be there. It's just that half of them would have been dead, or 3/4 of them would have been dead. Right, so whoever plotted that batch growth curve was counting the live cells, the viable cells. And the way they did that was probably by determining the CFU per mil by plating. So they would take a sample out every 5 minutes and they would put it on a plate, and then they put all the plates in the incubator, and they had to wait until the next day to find out how many bacterial cells there are. All right, well, that's not really practical because maybe tomorrow that's too late. So you might want to count the cells directly in a hemocytometer or directly using turbidity. But the problem is that counting directly in the hemocytometer does not tell you dead versus live cells. And counting using turbidity, the A600, does not differentiate between live and dead cells either, right? So the hemocytometer and the turbidity, in particular, the turbidity is not a reliable method of measuring how many live cells you have. It's a method of measuring how many cells you have, but it doesn't distinguish live from dead, right? So if I could ask you a question on the final exam about the shortcomings of each of these methods. So the shortcoming of the turbidity reading is that you are not sure whether you're counting live or dead cells. Okay, now the hemocytometer, there is a stain. There are a couple of stains you can use, like trypan blue, for instance, which will allow you to differentiate between live and dead cells. And we talked about that in the first week of class, right? So trypan blue will absorb into dead cells and live cells. But the live cells will pump the trypan blue dye back out. So a live cell will appear to be transparent, while a dead cell is dark blue, right? So the hemocytometer does have the ability to distinguish between live and dead cells if you stain those cells. If you don't, then you can't tell the difference. CFU only counts the live cells. It doesn't count the dead cells in any way. The turbidity meter, A600, doesn't distinguish between live and dead cells at all, right? So we have one method, hemocytometer, where you can distinguish between live and dead cells if you stain. We have another method that only measures the live cells but not the dead ones. And then we have a third method which doesn't distinguish between live and dead cells. So there's a hemocytometer slide. Once you spread the bacteria around, you have this, and look at it under the microscope, you have this grid that the amount of liquid that you put into the hemocytometer is precisely controlled by the cover slip. The weight of the cover slip determines how thick a layer of fluid you're looking at, and therefore how much volume you're looking at. And then this grid over here allows you to count the cells that are present without losing your place. It makes it easier to count them. Okay, so you count up the bacteria that way using the hemocytometer. Okay, colony forming units. Okay, so this is where you spread the bacteria onto a plate, you grow the bacteria overnight, and then you count the number of colonies the next day, being careful not to count plates that have more than 300 or less than 30 colonies. Okay, now, you might have heard the term streak plating versus spread plating. Here's a good question for you. What is the difference between streak plating and spread plating? And what is each technique used for? In fact, that is a good exam question as well. Okay, so what are these two techniques and what are they used for? With streak plating, what you do is you take a sterilized wire loop. It's a long wire. It's a wire with a little loop at the end. And you dip it into some bacteria, and then you streak the bacteria, literally like you were drawing a line with a pencil. You streak it across the bacterial plate, right? And then what you do is you want to streak the colony, you want to streak the bacteria in such a way that the bacteria kind of get separated from each other so that you can see a nice isolated colony. Because another way that you can identify bacteria is not just by the morphology of the cells, but by the morphology of the colony as well. I mentioned this during the yeast or during the fungal lecture, and that's that not only does every bacteria have a different shape, like some of them are rods and some of them are bacilli and some of them are lophotritious, vibrio, and so on, but the colonies that they form on plates are unique as well. Some of them are perfectly round, some of them have a jagged edge, some of them have a flat top, some of them have a rounded top. Some of them are gray in color, some of them are white in color, some of them are kind of orange in color. And so the morphology of the colony is also a way that people use to identify the bacteria. Right, so we have many ways that we can identify bacteria by looking at the morphology of the individual cells, by looking at the morphology of the colonies, by looking at the presence of genes when we're doing PCR, by looking at the nutritional requirements when we're analyzing them physiologically. Right? So the purpose of streak plating is to take a culture of bacteria and then spread it on a plate in such a way that you create well isolated colonies. If the colonies are too close together, they're competing for the same food, and then they end up being smaller than they otherwise would be. And so you want them to be nicely spaced apart, and that way you can tell the diameter of the colonies. That was in one of the assignments I gave you earlier this year. So you can tell the diameter of the colonies and what color they are and what kind of a border they have and whether they're rounded or flat on the top. So streak plating, the purpose of streak plating is not to count the number of bacteria that are present. The purpose of streak plating is to get isolated colonies and then look at the colony morphology. Also, if you have isolated colonies, you can be sure that that colony is made up of only one type of bacteria. Right? So if you get an isolated colony, that colony was generated originally by just one bacterial cell. Therefore, all of the bacteria that make up that colony must be clones of one another. They must all be the same type of bacteria. So if you have a mixture of bacteria and you're trying to isolate just one type of bacteria from that mixture, the way to do it would be to do streak plating. Right, so you do streak plating. That's another reason why you do streak plating. So you streak the colonies out, you streak the bacteria out to get well isolated colonies, and then you can take one of those colonies and use it to inoculate a broth culture, and you can be certain that that broth culture is only going to be growing that particular type of bacteria. Spread plating, by contrast, is where you take a spreader, a sterilized spreader, this thing that looks like a glass hockey stick, and you spread, you put a milliliter of blood or urine or something like that onto a plate, and then you spread it out to make sure the bacterial cells are well separated from each other. And then the main purpose is to count the number of colonies you get. And so streak plating is usually used to identify colony morphology. Spread plating is usually used to count the number of CFUs per mil. Okay, so here's how you do streak plating. You take a loop, a wire loop, and what you actually do is you sterilize it by holding it into the flame of a Bunsen burner. So you heat it up until it's red hot, and then you let it cool off. And when you heated it up until it was red hot, you killed and basically vaporized any bacteria that were on that metal loop. And then you dip the metal loop into a tube that contains blood or urine or sputum or something like that. And then you streak it onto the plate, and you do it in such a way that you will get some isolated colonies the next day. Now, the way that you streak it is very important because there's a technique to getting isolated colonies. And so if you're a technician who works in a microbiology lab, this is one of the things that you would need to know how to do. Okay, so what you do is you take the loop and you streak it on the plate like this, right? And then you take the loop and you frazzle it, you fry it again to kill any bacteria that are there, so it's sterile again. And then you take that sterile loop and you pass it through this area that you just streaked a couple of times, and then streak, streak, streak like that, right? And then you flame it again to sterilize it again, and then you pass it through that area that you just streaked again, and then you go streak, 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 streak like that. And then you sterilize it again, and then you pass it through this area again, streak, streak, streak. And so what's happened is that here in this area, there was a lot of bacteria on that loop, right? So then we expect that you won't have individual colonies there, which you don't. Right? And then this time we took a sterile loop and we just dragged a few of those bacterial cells out of that area where they're dense, and then diluted them a bit, and then we diluted them a bit more down here, and then we diluted them a lot more down here, right? So we expect the bacteria to be very concentrated there, and so we won't get any colonies. We expect it probably to still be very concentrated there, so we won't get any isolated colonies. But if we continue on, finally in this area, generally you will have isolated colonies, which you see. Right? And so notice how in this illustration is actually pretty accurate. Notice how the colonies are very small when they're packed together like that. And that's because they're all competing for the same food. They don't grow as big as they would if they were isolated. But if everybody has enough to eat, then they grow to their normal size. And so that's one of the ways you judge what bacteria that is, because the size of the colonies is one of the ways you identify them. Right, so this is the pattern that you use, and then this is a typical plate on the right. This is a typical plate that you would see. So the place where you initially streaked the plate in here, way too dense, and then over here in the third or fourth place that you streaked the loop, you have nice, well-isolated colonies. Okay, and here we had a mixture of different bacterial cells in the same liquid, so by streaking them out, you managed to isolate them from one another, right? So you can see that these white colonies are different from these dark colonies, but they were originally from a mixed sample. But by streak plating, you were able to isolate them. Let's look at spread plating. Spread plating is where you have a bacterial sample and you have yourself a Petri plate that contains LB agar or blood agar or MacConkey agar. You put a milliliter of liquid onto that plate, and then you take a sterilized glass rod and you spread that liquid around until basically it's absorbed into the LB agar. And then you put it in the incubator and you come back the next day and you count the number of colonies. And you assume that each colony arose from a single cell, and therefore that you're counting what you're actually counting is the number of colony forming units per milliliter, not the number of cells per se, because some of them were dead. Okay, so here's somebody using the glass spreader. You sterilize the spreader in between uses by dipping it in alcohol and then setting it on fire. You have to be careful when you're doing that because you generally have a jar of alcohol sitting there, and you spread the bacteria, then you dip the spreader in the alcohol, and then you have a flame there that you put the spreader into the flame, and it sets fire to the alcohol, and the alcohol burns off. And then you let the spreader cool down. You have to let the spreader cool down, otherwise you'll fry the bacteria when you try to spread it with a hot spreader. So here you see somebody doing exactly that. So they've got a spirit lamp here, and here they've got a beaker full of alcohol, and here's the glass spreading rod, and they've got a Petri plate in their hand. Right? So you dip the spreader into the alcohol, you set it on fire. When the fire goes out, you know the spreader is more or less cool enough to spread the bacteria, and then you spread the bacteria on the plate. But you have to be careful when you do that, because when you're going back and forth between a jar of alcohol and a flame, you might accidentally set the alcohol on fire, which I've actually seen that happen one time. Uh, Nobody got hurt, but a friend of mine was streaking about a dozen of these plates, and the alcohol flame is very dim, right? So you have this pale blue flame that you can't see, and so she kept on moving between the Bunsen burner and the alcohol without realizing that the spreader was still on fire. And so she ended up dipping a lit spreader that was on fire into the alcohol jar, and then the whole bench caught on fire, but nobody was hurt. So you have to be careful with flames, of course. Okay, so what about if you have too many bacteria? We have to do a serial dilution. We touched on this issue when we discussed virology. Okay, so too numerous to count. TNTC stands for too numerous to count. So we cannot count plates that have more than 300. Remember that. In order to avoid that, we want to count plates that are between 30 and 300. In order to make sure that we have some plates that have between 30 and 300, we do something called a serial dilution before we even plate these samples, so that the next day when we come back, we're guaranteed to have at least some plates that we can count because they're in the proper range. Right, so on the left, we have a proper amount of colonies. On the right, we have way too many to count, TNTC. Right, so what you do is you take your sample, and then you do a series of dilutions, and then plate each of those dilutions, and hopefully the next day you'll have some plates that you can count. Right, so you take your sample, and you put 1 ml into 9 ml of sterile LB broth, and then you mix it up, and then you plate 1 ml, and that constitutes a 1 in 10 dilution. And then you take some of this, and you take 1 ml of that, and you put it into 9 ml of sterile medium, and then you plate that. So that would represent 1/10 of a 1/10 dilution, or 1 in 100, right? So we took a 1 in 10 dilution of a 1 in 10 dilution. That's a 1 in 100 dilution, right? And then we do the same thing. That's a 1 in 1,000. Same thing. 1 in 10,000. Same thing. 1 in 100,000, right? So let's say that we had 30 colonies on this plate. In order to get back to the actual number of CFU per mil, we would count the number of colonies, 30, and multiply it by 100,000, which is the dilution factor, right? So that means 3 million CFU per mil, right? So that's the purpose of a serial dilution. If you get a job as a microbiology technician, or you go to microbiology graduate school, you'll be doing serial dilutions like that all the time, right? So it helps to know what they're useful for. Okay, so in this case, we have bacteria that are growing in the turbidity count. We're looking at a turbid broth culture, and the problem, as I said, is that you put this into a spectrophotometer, and it's counting both the live and the dead cells, because both the live and the dead cells contribute to the turbidity of the culture. Right. So the broth culture on the right is not turbid. The one on the left is quite turbid. Right, so you put it into a spectrophotometer, put it in a test tube, you put it in a spectrophotometer, it measures the amount of light that came through, and by extension it can measure the amount of light that was absorbed, and you can use that to figure out how many cells are present. You just can't tell whether they're alive or dead. Okay, now this is an interesting thing. This is a portable spectrophotometer used for measuring densitometry in the turbidity rather in the field. Densitometry is another term for it, but turbidity, right? And notice that there's a little round hole here that's covered by a cap. That little round hole is meant to receive a test tube. And now this over here on the right is an interesting type of a flask. It's referred to as a sidearm flask. Notice that it's kind of a combination between a test tube and a flask. Right? And so so what you do is you tilt this flask, allowing some of the culture to drain into this tube, and then you put the tube into the hole on the spectrophotometer, and you can measure the turbidity without having to open the flask. Right? So that's very useful if you're growing bacteria that's an obligate anaerobe, or if you're growing a bacteria that's particularly deadly, that's very dangerous, like tuberculosis, Mycobacterium tuberculosis, for instance, you might want to grow it up into large broth cultures, but you don't want to measure. You don't want to keep on opening the flask to take samples out and count them because you could infect yourself. So that is a sidearm flask, and the purpose of it is to allow you to measure the turbidity without having to open the flask. Okay, so here are typical growth curves. Okay, so to summarize the counting cells, we talked about the hemocytometer, colony forming units per mil, and turbidity. All right, so the whole thing that we discussed liquid versus solid cultures and the use of Agar to make semi-solid medium. We talked about selective versus differential media. We talked about batch versus fed cultures. We talked about autoclaves, orbital shakers, tube rotators, and rocker platforms. Talked about using dichotomous keys to identify bacteria based on physiology. We talked about using the PCR machine and barcode genes in order to identify bacteria that way. There are a few exercises here that you can try on your own time, right? So there are some questions, typical questions for quizzes and final exams. Here's a little exercise that I included where I have a whole bunch of pictures, and then on the right-hand side we have the names of all of these things, and so you have to identify what the picture is, match the picture to the name, right? So which one is this? That is an ELISA plate, isn't it? It's a microtiter plate or an ELISA plate, right? So this one is, the answer to this question is that, right? So there's several of them. What's this? I won't tell you the answer. What are these? What's this for? What is it and what's it for? What about these? What about this? What is it? What's it for? What do you do this for? What do you call this technique? What's this? What's it used for? What's she doing here? Okay, so I'll ask you, is she doing streak plating or spread plating here? And I think you know the answer. Okay, this is an interesting thing. This is a stir plate, right? And so it is a cheap alternative to an orbital shaker. So what you have here is a magnet in the bottom, and then there's a chemical stir plate. And what you did was when you sterilized the LB broth before you inoculated it, you sterilized it with the stir bar in there. So you don't have to put a dirty, non-sterile stir bar into the LB medium. It was sterile to begin with. Okay, so this is a problem like you had this is a problem like the one you had in the midterm. It's using the physiology of the bacteria to identify it, right? So this is something that you would do, identify bacteria using a dichotomous key, write the results. This would be a table that you would look up the answers on. Right? And then here's an exercise where you can match the terms to the definition. That the answers to these are in the problem set for this week's lecture.